So today we will be opening the doors of perception. I understand that's legal in Colorado now. Our first talk is going to be Jessica Cardin. She'll be talking on functional flexibility, state dependence in cortical circuits.
Well, it's always a pleasure to be at this meeting. It's always a wonderful mix of experiment and theory, and really an honor for me to give a talk here. And for those who aren't familiar with this, and I actually wasn't until like a week ago, in 1954, Aldous Huxley wrote this book called The Doors of Perception, and it was based on his experience in '53, where he took mescaline. And he sort of considered that the brain was sort of like a reducing valve, that it constricted our experience of consciousness and perception, and that by taking mescaline, he could open the doors of perception and really release his brain to process all of the available information about the environment, not just a restricted set of components. And I cannot promise, in all honesty, that I can open the doors of perception and explain everything about how perception works in the brain, unfortunately. But I will talk a little bit about both some of the evidence for how the brain processes information from the environment in different conditions, and also dive just a little bit into what we think the circuit mechanisms might be. And that is hopefully all legal in Colorado. So I want to start by saying that what we're really interested in here is the idea of functional flexibility in circuits. And that boils down to the idea that you have a physical set of neurons and synapses in your brain. And especially in the cortex, those circuits are tasked with an enormous amount of work. They have to accurately and repeatedly represent all the information coming in from the environment in all kinds of different places and under different conditions. And yet, you don't grow a whole bunch of new neurons and new synapses every time you encounter a new environment or learn a new cognitive task or encounter a new demand. Right? If you did, you'd be dragging around a brain the size of a house. Instead, there are elements embedded in those circuits that allow the functional circuit, or the neurons that are active and participating in any individual operation, to change on a moment-to-moment basis and to adapt very rapidly, even faster than synaptic plasticity on the time scale of milliseconds, to adapt to changes in the incoming flow of streams of information from the outside environment, or to different cognitive demands or different behavioral states. And that's a really large parameter space for us to try and get a handle on. And so what I'll tell you about is sort of ways in which we try to reduce this problem and to capture the circuits in the act of being flexible. We tend to use behavioral state change as our index, because it's something that we know we can capture in the lab, we can see repeatedly, and it's fairly tractable. And the idea here is that we'd like to capture the circuits going from state A to state B repeatedly, be able to fully characterize state A and state B, and identify all the things that happen at transition points as this flexible function, recruitment of different neural populations into a circuit at different moments really plays out in real time. Okay, so our, this has stopped working. Our circuit, I don't know if the computer has stopped working. Let's try this one more time. Nope, the whole thing froze. Let's try this again. So the idea here is to take a model circuit in the cortex, and for us, that's primary visual cortex. And here I'm showing you a highly schematized version of a visual cortex circuit, reduced to some pyramidal neurons, shown here on the right, and 3 major populations of GABAergic inhibitory interneurons. And we want to try to understand how the interplay between these cells changes over time. And I'm highlighting the inhibitory interneurons here because I will talk about them a lot in the second half of this talk. We think that interneuron diversity and the diversity of properties and synaptic targeting among these populations is really one of the key mechanisms or components of functional flexibility, and that potentially the recruitment of these cells into the circuit at different time points can help shift the circuit from one mode of operation to another. I've highlighted one population of interneurons here that I'm going to talk about quite a bit. These are the vasoactive intestinal peptide expressing interneurons, or VIP cells. They sit in a really interesting node in this circuit in that they receive a wide range of different kinds of inputs, but thalamic input, local excitatory inputs, long-range top-down feedback inputs, as well as very strong neuromodulatory inputs from several different systems. And so they are in an interesting position to integrate all kinds of behaviorally relevant information and then to influence the rest of the circuit. And these cells are a good example of a motif in cortical circuits that has really sort of come to the notice of the field a lot more recently, in that there's a lot of inhibitory to inhibitory interactions in cortical circuits. And we're just beginning to understand how these are important for both information processing and for cognition and perception. And I'm going to sort of highlight how we've started to unpack some of those inhibitory to inhibitory interactions in the context of state-dependent and visual processing in the cortex. I'm going to start from here. This is our standard behavioral paradigm in the lab. This is mouse on a wheel. And some of you have probably seen videos like this one before. So the animal's head is very stable as it runs on a wheel. And in this particular paradigm, all we're doing is capturing a very reliable, spontaneous behavioral transition. Mice will do this several hundred times in any given experimental session. And if you watch this mouse, he'll go from sitting to running to sitting to running, from a low arousal state or quiescent state to a high arousal state many, many times. In association with that, we and others have identified some internal markers of state change, like local field potential activity, and some semi-external markers of state change, like pupil diameter. So if you watch carefully here, you'll see that when that mouse is very quiet and unaroused, his pupil will constrict, and as he starts to run, pupil will dilate, and this is reflective of a number of changes in the brain, including changes in the level of neuromodulatory release for both norepinephrine and acetylcholine and a number of other things. Right. Now, the idea here for us, of course, is to use these state transitions as a way to capture the circuit being flexible in action, to see who's turning on and what happens to information processing in the cortex at different times. I think this process is really intuitive to anyone who owns a brain. So, for instance, after a late night at the poster session with some overpriced drinks, you might be sitting in a large lecture this morning, you know, a little zoned out, not really paying attention. Maybe you're doing the head bob thing, right? And you may hear the speaker talking, but you're not really processing that information, and you won't remember it very well later. However, if your neighbor suddenly elbows you a little bit to make you pay attention, you might sort of come to a very alert and attentive state of high arousal, and all of a sudden you're really listening to the words that the speaker is saying, and you'll have a good chance of encoding and remembering that information later. So this is something that happens to humans and to animals, and for what it's worth, all these biomarkers that I'm going to talk about in mice also are well mapped in humans as well. All right, so if we look at pupil diameter, we find that pupil diameter as a marker for arousal varies pretty nicely across different states. Here, sitting is in blue and running is in red, and we've normalized to the mean running pupil diameter here. And all this is meant to show you is that there's a nice wide parameter space of behavioral state through which we can track a cortical activity. Now, when we do that, we do it with a number of different methods, both pupil monitoring here in blue. Here we're monitoring locomotion of the animal in green as it moves on the wheel. We can also look at unit activity or local field potentials in the cortex, and later I'll show you some intracellular recordings as well as some 2-photon imaging. Then this is really capturing a well-documented, now well-characterized element of cortical state transitions, where as the animal becomes aroused, we see dilation of the pupil first. We see an anticipatory increase in firing rates in neurons, single neurons in the cortex, and we see a shift in local field potential activity from high-amplitude, low-frequency events to low-amplitude, high-frequency events, and a lot of gamma-range activity. Okay. So all this suggests that there is something transitioning in the cortex, and the whole mode of operation of cortical activity is changing. So you get a more detailed view of what happens in the cortical circuit. And I don't just mean like 1 or 2 neurons. I mean the whole laminar cortical circuit as the state of the animal is changing. We can do a series of other experiments. In this case, what we're doing is putting a laminar probe with recordings at evenly spaced sites all the way through the depth of the cortex to record local field potentials. And then we're putting in a pipette nearby to record intracellularly from one neuron. What that allows us to do is to calculate the current source density profile across the cortical layers. So this is the second spatial derivative of local field potential. What it gives us is a quantitative, I would say, estimate of the current transients associated with synaptic activity in different layers in the cortex. And in the example I'm showing you here, what we've done is we've taken the single spike events of the intracellularly recorded neuron and time averaged our current source density signal around them. That gives us a laminar profile of the current flow events, sinks and sources, that are associated or correlated with this evoked or spontaneous spikes of an individual neuron. And here you can see for this layer 2 pyramidal cell shown in the little triangle here, the spikes of this neuron are associated with a large current sink in layer 2-3. Not too surprising. Okay. However, we can take data like this and divide it up between behavioral states to try to understand how the pattern of activity across the cortex is associated with changes in the recruited activity of individual neurons. So here, sinks are in blue, sources are in red, and these are examples from 2 neurons, both in layer 3. And I picked these 2 examples because they are wildly different from each other, really nicely highlighting the diversity of neural activity and patterns of neural activity in the cortex. And here on the left, you can see this neuron when it's sitting. Its spikes are associated at time 0 in this dotted line here with a complex pattern of sinks and sources, mainly layer 4 and layer 2-3 activity. However, when the animal begins to run, the pattern of current sinks and sources associated with the firing of this neuron changes pretty dramatically, and you see that there is now an extra amount of activity here correlated with layer 5 and some in layer 6. In this other example on the right-hand side, what you should be able to see is when the animal is sitting, we see a very large extended sink that runs from layer 4 down through layer 5. When the animal starts to run, we see not only a change in the amplitude and the position of that pattern of activity, but also a change in the spatiotemporal pattern of activity across the laminar flow in the cortex associated with the spikes in that neuron. And these are just 2 examples. But they really highlight the idea that there is a remapping of the synaptic activity that's driving the spikes of individual neurons across the cortex when the animal changes from one mode of operation, one behavioral state, to another. Now, those really profound changes in synaptic activity and current flow in the cortex are strongly associated with modulation of visual encoding. This is sort of old data, but it really nicely highlights this point. So here what we're doing is recording from single neurons in visual cortex. We can see background activity as well as encoding of a visual response to a drifting grading stimulus. This is a very simple stimulus. On the right-hand side is quantification of the amplitude of the visual responses of these neurons across different states. And this is a signal-to-noise ratio index value. So excitatory cells, or putative excitatory cells, are in blue. Putative inhibitory cells, these are the fast spiking units, are in orange. And across all cell types that we've recorded so far, what we find is when the animals are aroused, shown here on the left-hand side as A, we see an enhancement of the signal-to-noise visual responses. When the animals become quiescent or a little zoned out, we see a reduction in visual encoding. Now, in correlation with that, which was really beautifully described by Marlene's talk yesterday, we also see that there's a state-dependent change in noise correlations. Noise correlations are considerably smaller when the animal is aroused than when the animal is quiescent. So overall, many streams of data like this have come together to give us a pretty good model of how we think changes are occurring in the visual cortex as the animal transitions from one state to another. As the animal becomes aroused, the pupil dilates. There are neuromodulatory changes in the cortex associated with that. The animal starts to become aroused or to run. We see an increase in the firing rate of neurons. We see an increase in the signal-to-noise and visual encoding. We know now that that those things are not necessarily related to each other. Those may represent separate mechanisms. But But it's important to note that this data and a lot of data like this represents a really unique state in the life of the animal. The animal's just sitting there passively watching a bunch of stimuli go by on the screen, and the stimuli have no valence for the animal at all. So what about a situation in which the animal cares a lot about the stimulus because the stimulus indicates a reward? In this case, what we've done is we've used a very simple go, no-go task. And we've picked this kind of task because we want to create a situation in which the animal is very free to cycle across a lot of different behavioral states. We want to see the full parameter range of behavioral states. We don't want to constrain the animal to be sort of very focused and very aroused and in one behavioral state all the time and always giving us responses. So we've picked a go, no-go paradigm, and the go stimulus is again a drifting grading. In this case, we've made the task a little harder because the drifting grading may vary in contrast, including stimuli that would be below the threshold for detection. The no-go stimulus is just a mean gray luminance screen. And the animal licks to a go stimulus, it gets water reward. If it doesn't lick, nothing happens. If it licks to a no-go stimulus, it gets punished with a timeout. So this is what the task looks like. The animal's sitting on the wheel. He waits for a stimulus to come on the screen. You'll see a drifting grading show up in a moment. He licks to get his water reward. He's successful in this trial. And then he settles down and waits for another trial to start. So it's a very simple, very straightforward task. However, what happens to performance across behavioral states is not so simple. So here, I'm not going to show you all the data from this particular project. I've just broken it down to some very simple numbers here. So in this case, I'm only showing you the easy stimuli. So these are the 100% contrast drifting gradings. This should be the simplest thing that mouse can do. Is just you can't miss the stimulus, except that sometimes actually the mice do miss the stimulus, in fact. When the animal sits, he gets about 70 to 75% of the trials correct. When he runs, he does just a little bit better. However, we weren't expecting this exactly. We found that the false alarm rates when the animal runs go up like a house. So he's getting a few more hits, but his false alarm rates have gone up tremendously, which means that overall his performance has degraded in this high arousal state where he's running or walking on the wheel. Now, this isn't just about motor activity sort of disinhibiting all motor responses, because when we do this in animals that are just trained to lick for water with nothing else going on, there's no correlation between locomotion or motor activity and the number of licks that they give. So there's something going on here where as the animal enters this the high arousal state where he starts to run, his perceptual performance is really degrading. So behavioral state is having a tremendous impact on the perceptual performance of the animals. Now, of course, we can go back and look at neural activity correlated with this. What we find is that it isn't so much the activity during visual stimulus presentation that turns out to be important, but the activity in the period preceding the visual stimulus presentation. So what we find is that in missed trials, when the animal is going to not respond to the stimulus, we see a lot more low-frequency, high-amplitude activity in the local field potential and cortex, a lot more highly synchronous activity, whereas in the HIT trials we really see the classic desynchronized cortical activity with lots of high frequency. We can look at this in more detail by looking at the spike responses of neurons across different layers. And again, I'm not going to show you exhaustive amounts of data from this project, but I'm going to highlight layer 5. So in this task, we know that layer 5 neurons are really important because they represent the projection streams of information leaving primary sensory cortex and going to key effector structures, in this case, the colliculus and the striatum. And we can break it down to layer 5A and layer 5B, which I've done here, which in visual cortex, there isn't a really good laminar separation between striatal and collicular neurons, but there's sort of a general distribution in that direction. In both layer 5A and layer 5B, here I'm just showing you the overall firing rates of activity before the stimulus ever comes on the screen. What we find is that when the animals are sitting, just knowing the spontaneous firing rate of the neurons is enough to give us about 85% accurate prediction of what the animal is going to do perceptually. So firing rates are elevated in layer 5A and layer 5B on hit trials, shown in these filled symbols, but not in the missed trials in the open symbols. However, so we can discriminate really, really well what the animal is going to do on each trial on a trial-by-trial basis just by knowing the firing rates in layer 5. However, all those values collapse into each other when the animal starts to run, and we can no longer see a difference between hit and missed trials. What about visual responses, right? This is a visual task. So here I'm showing you activity in the for period, the spontaneous activity on the left in each case, and then the visual response to 100% contrast drifting grading. So this is the easy stimulus again. I've split it into sitting trials and running trials. And when the animal is sitting, we do still see this disparity in the firing rates, baseline firing rates between hit and miss trials. But more interestingly, what we see in the hit trials here is that the animal has a very robust visual response to the stimulus that's not present in the miss trials. So when the animal is about to have a miss trial, it's not going to respond to this very high valence, high contrast stimulus. There's no visual response in layer 5. However, when the animal runs, we don't see any of these signatures. In fact, we don't really see visual responses during this task when the animal is running during trials. And there is no difference between hit and miss trials here. So this kind of change in cortical activity that we see between quiescence and arousal, between sitting and running, those changes have profound implications for what the cortex is actually doing with information from the environment on a moment-to-moment basis. They can regulate, those changes can regulate the efficacy of encoding, noise correlations change in the correlations between neurons, and we see changes in the activity overall of cortical neurons, but we also see dramatic changes in encoding associated with behavioral state. Okay. So that was pretty much all by way of trying to convince you that this kind of behavioral state change is really important for the cortex, right? Changes a lot of things about how neurons interact. And what I'm going to tell you about next is some data where we're just hitting the tip of the iceberg and trying to unpack the circuit-level mechanisms that might underlie these very robust changes in activity in the cortex as the animal changes state from moment to moment. And I've put the schematic of the cortex back up here, and I want to re-highlight the idea that GABAergic interneurons, because they are very diverse in their properties and their synaptic targeting, may be really a key element of this state-dependent regulation of cortical activity. So we and others, there's many papers now, have identified that there is a state-dependent recruitment of different populations of inhibitory interneurons, and in particular these VIP interneurons, which represents somewhere on the order of 1% to 2% of all cortical neurons. They're heavily regulated by behavioral state, and they're recruited at the onset of locomotion. So that seemed to us to be a good enough reason to try to use them as sort of the entry point to trying to unpack these cortical circuit interactions. And we've done that in a number of different ways, and I'm going to hit the highlights of 3 different ways that we've looked at manipulating the circuit to understand not only what the VIP interneurons might do to cause a shift in the mode of cortical operation, but also how they might be impacting other inhibitory interneurons in the local circuit. Okay, so in this first set of data, what we've done is to take advantage of some molecular genetic techniques. And in particular, there is a tyrosine kinase receptor, and I won't go into the details, which is really important for the development of excitatory synapses onto inhibitory interneurons and synapses from those inhibitory interneurons onto their targets. And so by mutating this receptor or deleting it from a target interneuron population, we can essentially turn down the gain on those cells. They're still in the circuit, they still have some activity, but their influence has been markedly reduced. And so what we've done in these experiments is to remove this protein from the VIP interneurons sort of around the second postnatal week of life, and then look to see what the impact of that manipulation is on the circuit, both with physiology and with two-photon imaging. And the idea here is just to start at the grossest possible level and try to figure out, do these cells really play a key role in regulating the circuit? And this is not a trivial point, right? There are not very many of these interneurons. They are like raisins in pudding, right? And so it would be, if we find that they have a really profound impact on the network, that suggests that there is something really important about the way that they're wired up to other cells, because there aren't enough of them to sort of control the cortex without some very special properties. So when we make this early mutation in these cells, we've sort of taken these cells functionally out of the network. What we find is actually that firing rates in the cortex start to go up. So excitatory neurons in the cortex, in the absence of really normal VIP interneuron activity, start to rise, and they start to rise by the adolescent period here, and they rise even more when the animals become fully mature. And I really should go back to this schematic for a second and re-highlight the idea that the VIP interneurons don't just synapse on other interneurons, the somatostatin cells. They also make extremely powerful synapses on the distal dendrites of pyramidal neurons. And so there is an open question here of what the functional balance or the role of those two inputs might be, since these interneurons may be simultaneously inhibiting other interneurons and also inhibiting pyramidal cells, leading to a bit of a conflict here where they might be disinhibiting pyramidal cells through the somatostatin cells, but also at the same time inhibiting the pyramidal cells directly. Okay, so one possibility for this increase in firing rate here that we see in the absence of VIP cell activity is that we've removed this distal dendrite inhibition directly onto the pyramidal cells. At the same time, we find that decreasing the influence of VIP interneurons in the cortex has pretty profound consequences for other elements of cortical function, not just firing rates. So these are responses to drift and grading visual stimuli again. In this case, I'm showing you responses that have been normalized to the baseline firing rate. This is population averages here. So baseline would be these two dotted lines. And what we find is that overall visual responses in pyramidal neurons in the cortex are also highly diminished. So firing rates have gone up, but visual responses have gone down. If we measure orientation selectivity, we find that it's pretty normal right up until that mid-adolescent period, where the mutants, sorry, I should have said this, the controls are always in black, and our mutated mice are in blue. So our mutants show a decrease in orientation selectivity that persists well into adulthood. Okay. So disrupting the VIP interneurons has disrupted not only firing rates of excitatory neurons, but also some very core features of visual encoding in the circuit. And if anyone's interested at the end, I'll be happy to talk about the rest of the things that are also messed up in the circuit. Now, we've, obviously by doing a mutation all the way across the brain, we've mutated cells that aren't just in the cortex. There are VIP positive cells in many, many other places in the brain. However, if we re-express the protein that we've deleted genetically, by putting it back in a virus and putting it back into the VIP interneurons, only in the cortex, we find that we can completely rescue all of the effects of the VIP interneuron manipulation. So rescuing only the cortical VIP interneurons completely rescues the changes in firing rates that I showed you before, where in our mutants we see this increasing elevation in firing rate of the pyramidal neurons. If we rescue the VIP interneurons, we find that firing rates never develop these changes and map onto our controls exactly. That rescue also restores visual function to the cortex. So in our controls, we see normal visual responses to drift and grading stimuli. In our mutants, they're significantly reduced. In our rescued mutants, they're restored to normal amplitudes, as well as the orientation selectivity of pyramidal neurons in the system. So all of the changes that I just described really, truly are the result of taking these very sparse interneurons out of the cortical circuit. Okay. So what about consequences for perception? Right, I've just suggested there are changes in firing rates, there's changes in visual encoding. Does this translate to a functional impact on the animal's ability to perceive stimuli out in the world? So to answer that question, we returned to this go, no-go stimulus paradigm that I described before. And it's a very simple task, contrast modulated drifting gratings. And one of the things that's interesting about this task is that in the learning phases, it behaves very much like a perceptual learning paradigm. And I'll describe the data for that in just a second. So the animals, as they learn this task, they actually become better at it in that the threshold for contrast detection decreases over time, usually over the course of about 6 to 8 days. In this particular task, we find that the very easy part of the task, the animal's ability to detect an incredibly strong high contrast drifting grating, doesn't actually require visual cortex. So the very easy parts of this task require only things like the colliculus. However, the harder part of this task, the detection of low contrast stimuli, the difficult trials, and also the animal's ability to learn the contrast varying part of the task, those elements of the task do rely critically on primary visual cortex. And so when we look at our normal control mice, so here I'm dividing their training sessions into the first few days, so probably I think this is the first 4 days of training, versus late, the last 4 days of training. So you could translate this as in early stages the mouse is still a novice, in late stages he's an expert. We find that the psychophysical curve for this task, so contrast is on the x-axis, hit rates on the y-axis. Even in the early phases, the animal does okay, or it does okay at this task. But as the animal becomes an expert, the whole curve shifts left, and the contrast threshold for detection decreases tremendously. For what it's worth, this works on humans too. So the undergrads in my lab have psychophysical curves that lie right on top of the mouse curves. One of the nice things about contrast detection is that your psychophysical abilities and the mouse's psychophysical abilities are extremely similar, as long as you control for things like spatial acuity. However, in our mutant mice, where we've interrupted the activity or the firing of the VIP interneurons, what we find is that the mice don't ever show this V1-dependent leftward shift, the decrease in contrast detection threshold. They always look like novices. So they can do the task, they can detect some of the high contrast stimuli, they can do all the motor actions that are necessary, but they never show this perceptual learning. Okay. So interrupting the VIP interneurons in their normal function has consequences not only for neural activity, but also for behavioral perception of visual stimuli. Okay. So back to our schematic here. So what we know from slice physiology data that I'm not going to show you here, that one of the things that we've done by mutating this particular receptor in the VIP interneurons is that we've removed a fair amount of the glutamatergic synapses onto the VIP interneurons. They never form. And we wanted to look more closely at the effects that this would have on the VIP interneurons themselves, not just sort of the aggregate effects on the cortex as a whole. To do that, we turned to two-photon imaging because these little suckers are small and they're really hard to record from with traditional electrophysiology. So this is a series of experiments where we've just put GCamp in the VIP interneurons. Here you can see a movie. The bottom traces the animal's position on the wheel. You can see as he starts to move, the VIP interneurons all turn on kind of simultaneously. When he's sitting still, they sort of fade into the background. So one of the interesting things about the VIP cells is that the whole population is modulated somewhat simultaneously by these state transitions and locomotion. Right, so in normal mice, and this has been shown very nicely by Mike Stryker's lab among others, in control animals at this locomotion time point shown in the white dotted line, all the VIP interneurons generally turn on very robustly at the onset of arousal or locomotion. However, in our mutant animals where we've removed the glutamatergic inputs to these cells, we see that this response is much reduced. And that tells us two things. One, this locomotion or state change-induced activation of the VIP interneurons is dependent not just on their cholinergic inputs, but also on glutamatergic input. And two, this may be one of the reasons why the circuit is so disrupted. The VIP interneurons just aren't turning on at the right moment. So we went and looked at state changes, state-dependent regulation of the circuit in our electrophysiology recordings. And this is just an example from normal mice. This is very robust. We've published it, others have published it. It's very consistent across mice and across layers. We see an increase in firing rate at locomotion onset time points, followed by a bit of a plateau, and then a decrease. So there's an example cell on the left and population averages on the right. In our mutant animals, we never see state transitions. These animals seem to have only one cortical state overall, and we never see any kind of influence of changes in behavioral state in either direction, either from quiescent to aroused or aroused to quiescent. That's highlighted here on the right-hand side. We've quantified this as a modulation index, which is robust in our controls in black and pretty much absent in our mutants in blue. Now, I should say, I don't actually know what state these animals exist in. I don't know whether they're always in a quiescent state or always in an aroused state, or if it's some other pathological state that's been induced. We just know that they don't have any kind of state transitions. Okay. So loss of the VIP interneuron activity and the sort of state-dependent recruitment of those interneurons into the network is associated with a loss of state-dependent transitions across the cortical network in the pyramidal neurons. And as I highlighted before, one of the most interesting things that the VIP interneurons do is to inhibit these intermediary interneurons, the somatostatin cells. The somatostatin cells are an interesting population in their own right, and they receive not only inhibitory inputs from various other interneurons, as well as lateral excitatory inputs, especially in layer 2-3, which is where the data I'm going to show you come from. They also are heavily regulated by neuromodulators, including cholinergic inputs. And so this interaction between state-dependent changes in neuromodulators and excitatory inputs and inhibitory inputs gives us a very complicated system with a lot of parameters. However, in our slice data, which I'm again not going to drag you through, we know that our manipulation of the VIP interneurons also removed the inhibition from the VIP interneurons to the somatostatin cells. So we can look again at these mice and ask, what do the somatostatin cells do in a context where they're not receiving VIP inhibition? To do that, we used an intersectional genetic approach to manipulate the VIP interneurons while imaging simultaneously from the somatostatin interneurons. So here we've taken our CRE-dependent mice and used them for the mutation that we induced, and we've crossed them to a somatostatin flip line so that we can express a flip-dependent GCAMP specifically in the somatostatin cells in the cortex. And that's what this movie shows here. So these are somatostatin cells, and here I'm just showing you the response to the somatostatin cells to visual stimuli because these are just beautiful. The whole population really responds robustly. Come on. There we go. Okay. Now, so the imaging data that I'm going to show you here is from layer 2-3. In normal animals, we generally find some heterogeneity of the somatostatin cell responses to arousal or locomotion. In control animals, we find somatostatin cells that are both positively and negatively modulated by locomotion onset, but on average, we don't see much aggregate positive or negative regulation. We generally see on average that the population as a whole doesn't show much modulation. And that's highlighted here on the left-hand side where we have individual somatostatin cells, some going up, some going down, and again the white dotted line here is the onset of a locomotion state transition point. Now, in our mutant animals where we've removed the influence of the VIP cells on the somatostatin cells, what we see revealed is a state-dependent recruitment of the somatostatin cells into the network at these same locomotion time points, suggesting that the somatostatin cells under normal conditions really exist in this balanced state where they do get some positive inputs correlated with arousal or locomotion, which are balanced out potentially by the inhibitory influence of the VIP interneurons, and that some titration of the relative influence of those inputs might lend a lot of control over the sort of subtlety of the recruitment of the somatostatin cells. I'm not going to show a lot of direct data on that right now. But we do see that in the absence of VIP input, the somatostatin cells become much more active when animals become aroused or start to run. Okay. All right, so all the data that I just showed you is very consistent with a really key role for VIP interneurons. However, it's also all the result of a sort of early postnatal developmental manipulation of those cells, and that leaves open the possibility that what I've just described is mostly about VIP cells having some very specific role in a developmental window for pruning or maturation of the system, as opposed to their adult role in the mature system. And to sort of examine the difference between those two things, we really need a different set of tools. In the next set of data that I'm going to show you, what we've done is to take our same approach with two-photon imaging, but in this case we are and some physiology, and we're expressing the potassium channel Kr 2.1 in VIP interneurons, and then looking at the activity of local pyramidal neurons. And we've done this, we sort of sliced up the developmental trajectory of the cortex by injecting a virus carrying the Kr 2.1 at 3 different ages, P2, P10, or P50. It's important in interpreting this kind of data to remember that there is a bit of a delay from, say, injecting a virus to peak expression of the protein. So you sort of have to add a week to 10 days to the time point to really get to the time point where it's probably having its biggest effect. So an injection at P2 would probably have its biggest effect starting at the end of the second postnatal week, or P14. Likewise, an injection at P10 would start to have its effect in that mid-adolescent period in the P20s. Okay, but in any case, we're injecting at 3 different age points. We're letting the mice grow up to adulthood, and then we are using mostly physiology to assay the function of pyramidal neurons in the cortex. And this is what we find. So if we turn down, so overexpressing potassium channel hyperpolarizes the VIP interneurons. It causes them to be considerably less active. They don't fire very much. And what we find is that if we do this in the early postnatal stages, this is a firing rate density histogram for control animals in black and our Kr 2.1 injected animals in green. We see a rightward shift in this distribution, suggesting an overall increase in firing rates, very consistent with data that I just showed you previously. However, if we do this at later time points, if we do this at P10 or P50 in the adult animals, we don't see any change in mean firing rates in the excitatory neurons. Suggesting that this sort of control of pyramidal neuron firing rates is something that's really a postnatal developmental role of the VIP interneurons and not a mature role for those cells. Then we can do the same manipulation and look at state-dependent regulation of activity in the cortex. We find in our control animals, obviously, we get a big modulation of activity in the cortex. If we look at our animals that were injected at this very early time point, we see in green on the left-hand side here a complete loss of state-dependent modulation of cortical activity, exactly as we saw in our mutants that I just showed you in the blue data. However, we get diminishing effects of this if we do this long-term manipulation later at P10 or in the adult animal. Now, one caveat to looking at this data, I should say, is that this is data that was compiled by collapsing across all our recordings across all of the cortical layers. So this includes data from layer 2, 3, layer 4, layer 5. And the error bars here, especially in the adult case here, are quite large. So although I'm not unpacking the data fully here, it's entirely possible that these effects in the adult animal are a bit variable across the cortical layers. And that's something that we're still looking at. And again, we can look at visual responses in these animals where we have manipulated the VIP interneurons to be very quiet. So when the VIP interneurons are silenced early in postnatal life at this P2 point, we see that there's no visual responses left. And in fact, we actually see a very small, just barely significant suppression of activity with the onset of visual stimuli. And again, these are all tested with drifting gratings. However, when we do these manipulations later in development, we really don't see any effect on visual responses at all. Okay. Now, we really wanted to go back and do this and look at the effects not just on pyramidal cells, as we can for their physiology, but on effects of other inhibitory interneurons. And again, because even the somatostatin cells are relatively sparse, this is hard to do with physiology. And so we had to switch approaches here to do this kind of manipulation of VIP cells and look at their targets, the somatostatin cells. So this next set of data And instead of outright killing the VIP, instead of suppressing the VIP cell activity by overexpressing a potassium channel, instead we're just killing them with a caspase virus. So in these experiments, what we've done is to go in to our, sorry, wrong animals here, just VIP Cree animals. What we've done is we've gone in, we've expressed a Cree-dependent cell death mechanism using caspase, and then we have we've expressed a flip-dependent GCAMP in the somatostatin cells in the same animals. And then we're using 2-photon imaging in our head-fixed prep to look at the somatostatin cell activity. So what I showed you earlier was that in control animals, our somatostatin cells tend to show a few cells with positive modulation, a few cells with negative modulation. In these animals where we've just killed off all the VIP interneurons in a local area, what we find is that our somatostatin cells are inappropriately recruited now at locomotion onset time points, very consistent with the genetic data that I showed you earlier. So in the absence of VIP inhibition, somatostatin cells show this state-dependent recruitment overall. And this is just distributions of modulation index for individual neurons. So in our control animals, this sits around the 0 point, some positive and some negative. The whole distribution is shifted to the right in our animals where we've killed off the VIP interneurons. And this is work in progress. If this is on Katie Ferguson's poster last night, if any of you saw it. And we're sort of continuing to do these point-by-point manipulations of the circuit to try to understand the dynamic regulation of the interactions between the different interneuron populations. Okay. So just to kind of wrap this up quickly, what I showed you was an initial series of data highlighting the idea that behavioral state is one key regulator of cortical function, that state-dependent changes in circuits really shift the activity of neurons, the visual encoding of those neurons, how the neurons send signals out to target structures, and also the animal's ability to perceive and respond to a stimulus that has some valence. Those things are all heavily regulated by behavioral state transitions. And we can use those state transitions as a hook, basically, to try to unpack how cells are recruited into the network, what neural interactions are happening at different times. The second part that I showed you was our attempts to start unpacking exactly what those recruitment events of different inhibitory interneurons look like, and how the interactions between distinct interneuron populations might really play a role in regulating this state-dependent visual encoding in the circuit. So we know that the VIP interneurons play a really key role both in development and in the mature cortex, partly potentially through regulating the pyramidal neuron activity directly, but also in large part by exerting state-dependent inhibition on the somatostatin interneurons. And overall, this suggests that we probably have only hit the tip of the iceberg. I expect that we're going to find many more of these complex state-dependent interactions between elements of the circuit. And I should point out here, as a plug for the theory side of things, that this is really where we experimentalists need good theory, because we can't test every single one of these things at the same time in vivo. Okay. Now, since I didn't do any of this work, I'm just going to highlight the people who actually did the work that I just told you about. So the initial state-dependent work was done by Martin Vink and Renata Batista Brito. Some of the VIP mutation work was done by Renata and published last year. But all of the unpublished data that I just showed you, the imaging and the interneuron manipulations were done by Katie Ferguson. And the behavioral paradigm work and state dependence was done by Quentin Paranode. And I will say that I am extremely fortunate to have amazing postdocs in the lab who are capable of doing all these things. And of course, we rely on our collaborators and funding sources as well. And thank you.
We have time for a few questions.
You showed very convincingly that state changes matter for perception. I was wondering if you could speculate on what happens to low-level properties of these neurons, receptive field properties. What's happening when these change, the state changes? Are the receptive fields different?
So I don't have a complete data set for this. But the bits that we have suggest that yes, there are receptive field properties that change with state, and you really only see that powerfully if you map the receptive fields themselves. It doesn't tend to show up very well in things like orientation selectivity index measurements.
Thank you.
Hi. Great talk. Thank you. So when I think about state transitions that are driven by interneuron networks, I usually think about it reflecting as different oscillatory activity happening in the network. So, and particularly with your current source density recordings, those have been used in the hippocampus to show a lot of the mechanistic underlying of these oscillations. So I was wondering if you had any comments on what these interneurons are doing in generating rhythms and what the participation is in the task.
So the cortex is not as beautiful a system as the hippocampus, unfortunately. We do know in this kind of task, we know that especially in that 4 period before the stimulus ever comes on, we see a dramatic shift in gamma oscillation activity, and we see a real counterbalancing of sort of delta range activity versus gamma range activity in that 4 period. We don't see a lot of really clean evidence for, gamma frequency being a key carrier of visual information, but this may not be the right task for that kind of analysis, to be honest. On the sort of mechanistic side, we have good evidence. Hillel Desnick's in the lab has done a beautiful job of showing that, gamma activity in the cortex relies not only on parvalbumin interneurons, but also somatostatin interneurons. We now have some evidence that the VIP interneurons actually play a role in a developmental and a mature context, potentially, in sort of setting up the balanced wiring that contributes to letting the circuit be able to do those things. So there's a lot of interactions here.
Great, thank you.
Would our next speaker come up while Jess answers a few more questions?
Hello. So I was interested in the critical period aspect of this disruption to VIP, and I was interested if you had any analogies to human disease, particularly schizophrenia, since that seems to have a number of similarities to the data you presented.
So everyone who hates molecular biology can now shut their ears. So ERB4, the tyrosine kinase receptor, is actually has been highlighted in a couple of recent GWAS studies, and was an old candidate risk gene for schizophrenia, in fact. In part, probably because it's expressed highly in both parvalbumin cells and VIP cells. And there's some good evidence that it participates in regulating, their engagement of PV cells in attention-related behaviors. So there's clearly a disease gene link here. And the window in which those manipulations had a really big impact on development is a really interesting window because it's an earlier window than the sort of classic parvalbumin plasticity window. And there's some emerging evidence that the different sets of inhibitory interneurons are important for somewhat overlapping but partially distinct developmental windows in that early postnatal period.
One more question.
Thank you.
Thank you for the inspiring talk. In your experience, out of all of these metrics that give you an idea about state changes, running, pupils, LFP, which one is the one with the highest predictive power that a state change is happening, especially on single trial, if you don't have the benefit of averaging?
So this, I think, is a really complicated issue, in part because in our hands, it's different when the animal is doing a task versus when the animal is just sitting passively. So when the animal is sitting passively, we get equal predictive power from locomotion and pupil diameter. When the animal is doing a task, it gets much more complicated. Pupil is highly predictive when the animal is sitting still, but when the animal is running, pupil is not very predictive, for us at least, of performance. So that may represent a really complicated relationship between neuromodulatory state and cortical or brain state.
Thank you very much.
All right, let's thank the speaker.